So I'm going to give a brief overview of the type of data that's available, connectomics data that's available already for Drosophila melanogaster. So this is just a brief overview of the structure. So I'll give a brief overview of the connectomics data, and then followed by David, and then Philip, and then we'll go on to our groups. The first thing to think about when we think about electron microscopy connectomics, which is what we're talking about here, is that we have both the shape of neurons and connectivity. And so we're taking, we're building up a shape of a neuron using features in the image data from the EM and putting it together into, in this case, a neuron skeleton that represents, I know, faithfully the shape of a neuron, including connectivity, so we can recognize both input and output synapses. And right now there are sort of three main data sets already. So the earlier one was a larval L1 connectome. So this contains around 10,000 neurons. It's now close to 100% complete. This figure is from 2017. This was led by Albert Cardona's group, and they've been reconstructing neurons on this since 2013, so quite a long time. And so I know there's some kind of stats on here in terms of the length of neurons and number of synaptic connections. And this is just more to contrast to the other data sets that we'll see, which are now from the adult Drosophila. And in this case, there are around 100,000 neurons. This data set, as we have it called FASB, has been reconstructed by a community of 27 labs. And you'll see that instead of 2 meters of cable, we now have 10 meters of cable. And so this was all, for the most part, manually reconstructed. And I will come to why that's important. The one that's most recent, which you might have heard of, is the HemiBrain, so generated by Janelia Research Campus. It was released in January 2020. And this is what we might call a dense connectome, and I'll explain later what this means. But you'll see just by looking at the number, for example, the length of the cable, number of neurons and synaptic connections, these numbers are now sort of much bigger than they were before. And so all of these data sets and data are available. L1 FASB are available through VFB CatMade instances as well. And I'll explain briefly what CatMade is in a couple of slides. And so you get access to the actual EM data and reconstructions of neurons, as well as having access to this data that's integrated into the main VFB site. And David will talk a little bit more about that, how that data is integrated. The HemiBrain data is also included in the Virtual Fly Brain site, but Janelia also has their own resource, which is called Newprint, which you can access through the link. And so how do we actually reconstruct neural morphologies? Like I mentioned, we can use features in the image data as humans to follow processes of neurons from section to section, and also to recognize what synapses are. And here, for example, we can recognize neurotransmitter vesicles, postsynaptic densities, T-bar, and so on. When we started out initially, and also the L1 project, tracing was done manually in CatMade, which is a web-based platform for reconstructing neurons and starting some analysis. So it's quite powerful for analysis as well. And in that platform, neuron shapes are represented by skeletons, what we call skeletons. So we would lay down nodes that follow the profile, of a neuron from section to section, and these nodes get connected together by lines. And there are different types of nodes for indicating what presynapses might be and what a postsynaptic partner would be. And so this was all so that in the end you can build a skeleton representation of a neuron. So a soma is just an added on sphere that represents the size of the soma. And so we can have a faithful representation of inputs and outputs and the shape of a neuron. So there have been a lot of developments in this area. And so now there are quite a lot of techniques and techniques that are efficient enough that they can be used in bulk to auto-segment neuronal profiles from EM data. One of these is flood filling networks developed by Michal Januszewski at Google. So this was the one, for example, used in the HemiBrain and for an auto-segmentation of FASB as well. And so all of these are machine learning algorithms. They use the voxel features in the image data to try and build neuronal processes through the image. One of the things that is worth mentioning is that each of these algorithms requires some training data, and putting together that training data is, is quite a lot of human effort to do. So annotated data, depending on the quality of the image data. So sometimes if it's a very big volume, like in the HemiBrain, the quality is not homogeneous from region to region. And so training data from one region might not work very well to recover neurons in another region. And then also there are decisions that need to be made in terms of how you balance the algorithm decisions to either have the longest neurons that you can have and accept that there might be some errors in there, that it's merging things that shouldn't be merged, or that it should stop as soon as a confidence level is below a certain threshold. And so this is kind of what we see here. And so we want to set that balance correctly so that we have the least amount of proofreading effort. And in general, splitting things that shouldn't have been merged takes longer than merging things that are just not merged. You'll see here there are other types of errors as well. So for example, very common is to have glia merged in to somas or to different parts of the neurons. And so one thing that will be quite important in the workshop as well is to understand how neurons can be represented. So usually in these automated outputs, you will get neurons represented as a 3D mesh, so a volume. And so you can see just by looking at it that there are certain morphological features that become really apparent. So we can see the dendrites are quite spiny, whereas the axonal terminals are quite bulbous. And that's not something that we can immediately see just by looking at a neuronal skeleton, as we had traced it in CatMate, for example. But the advantage of the skeleton is actually that it's really flexible to do a lot of the analysis. And so some of the analysis that, some of the examples that you follow in the workshop, you will be using neuronal skeletons that have been generated from neuron meshes. And that way, I know it makes it very easy, for example, to subset a neuron. So to take just some part of its arbor to do some other analysis. One thing to mention is that sort of, dendritic inputs to a neuron. So we can divide kind of a neuron arbor into two main types. So one is the large profile neurites, which we'd call the backbone, and the very fine neurites, which is where actually most of the dendritic input comes in, which are called the twigs. And just because they are very fine and hard even for humans to trace, it means that even these automated segmentation algorithms, they're not recovered very well. And so I know, so we can do this. Does it actually make a difference in terms of our output? And the answer is yes. So it makes tracing and reconstructing neurons much, much quicker. So one particular example is this flood filling network segmentation of the whole of FAFB, so which is the one, this FFN1, in which we're comparing some semi-automatic reconstruction of neurons with a fully manual one, and this gives us a speed up of 15 times, okay? And that's really significant. And even so, in these neurons, one example of this is that we still get neurons that are made up of many fragments, so we still need to put them together. But even then, that is just by itself much quicker. Other types of segmentations might give longer fragments of neurons. So one of them is the Flywire project, and this is from Sebastian Sung and Malamurthy's groups. So there is also a community and a web platform where people can trace FAFB. So they realigned FAFB and then ran another auto-segmentation algorithm. And then just by the fact that the data was in better quality, and so align better, the auto-segmentation gives better outputs. And so this is the same neuron in the two algorithms. But even then, even though this neuron looks, quite complete, there's still quite a lot that's missing. And so you always need proofreading on top of that. So the other part that we need, so we have the neuron shape, but now how do we get connectivity in bulk? And so we can also use auto-segmentation methods for synapses. So one of the more recent ones is Jule Buhmann's method from Jan Funke's lab in Janelia, in which she ran a prediction of synaptic partners in the whole of FAFB. So you can see here 244 million synaptic partners. There are quite a lot of false positives on this, so the results need validation. And so this is one of the things in all of these auto-segmentation methods, that there needs to be some additional proofreading. The other method, or slightly different method that was used in the hemibrain, for example, was it also auto-segmented T-bars independently from PSDs. And so here you're just seeing an example of how they look in the EM and on a 3D. So each link is one connection. And so in general, these methods can be tuned. And for example, in the hemibrain, for PSDs, these methods don't do significantly worse than humans would do in identifying PSDs. But there is quite a lot of variability in terms of how much they will recover. So T-bars are quite big and very easy to spot even for humans. And so there's a high recovery rate for those, whereas PSDs, which are quite small and can be, very different as well in terms of how strong they are. You can get very broad range of recovery for these. And as I mentioned previously, depending on the quality of the data in a particular region, you might need to tune the algorithm to do particularly, to improve your recovery on that region. And so now we've got the output of this auto-segmentation of neurons and synapses. And so now comes the proofreading, which is actually takes quite a lot of time. So for the hemibrain, it took 50 persons year of proofreading time. And all of this proofreading means that the more you proofread, the higher quality the data will be. And just to mention, I know we're talking about incomplete arbors of neurons. This always is directly correlated to incomplete connectivity. And so in the proofreading, there might be large mistakes, so false merges that happen. In this case, two of these cells are merged here because these two profiles got merged together. Or you can have some glia that is merged together onto one of the branches. You can also, the other part of proofreading is also to complete your neurons to a particular level. In this case, you have this branch and we're adding these 3 others that were missing, and there'll probably be many more missing there. But the level of completeness of the data will depend, particularly if it's a very big project, it will depend on the release goals of that, or if it's within a group, it will depend on what you need for the analysis that you want to do. And just to mention, I know completeness, when we talk about completeness, it might mean that one part of a neuron was done to completion. It doesn't mean that the whole neuron is done to completion. That's, and of course, when we're thinking about why do we want to complete neurons, is that the more complete the neurons are, the easier it is to identify cell types and to distinguish neurons from each other, and to identify new cell types if they are not known. So I mentioned previously dense connectomes and what this means. And so when we talk about a dense connectome, just means that every neuron in that region or in that volume is segmented. So this is usually the output of an autosegmentation method. So there's always a caveat. So when I say every neuron, just means every neuron above a certain size threshold, because there will of course be many small fragments which are not attached to a neuron in there. And again, this relates to the completeness that will be required. So in the hemibrain, for example, this is a plot of the neuropiles on the X and the completion on the right. And by completion here, they mean the number of postsynaptic densities that are attached to neurons. Okay, so the more postsynaptic density in that particular region that are attached to neurons, the more complete it is. And you'll see some of these neuropiles that are the most complete, which are around 80%, just over, are some of the smallest. And this is because there was a lot of a lot of proofreading effort put onto those. And so when we're proofreading, we're adding arbor, but so we're also adding synapses. And here's just an example, again, from the hemibrain of sort of synapse. So 30% completeness versus 50% completeness, and how many synapses are added after proofreading and how many synapses are removed. And so in general, you're adding synapses to the data. And so that's good. So, but it also means there are a lot of them are missing. And so if a lot of this is missing, is the data still good enough to work with? And the answer is yes. So we know that for the most part, there is a very long tail of weak connections in the data. And even though on average in the hemibrain, we have 40% recovery of synapses, we can clearly distinguish strong partners. Okay, so we will get definitely strong partners from the data. We won't get every weak partner, but these are probably, many of them will be random, just making 1 or 2 synapses. I've kind of mentioned completeness in different contexts so far. And so just to make it very clear, there are different ways of defining completeness. So one might be related to the volume. And so the hemibrain, we knew it was a truncated volume. And so you can see it in green here, what it contained. And so of course, the neurons that are in a truncated volume will be truncated. And so you don't get the full morphology of a neuron, which will make it harder to identify what that neuron might be. The other type of completeness might be of what I'm calling morphologies. So are all neurons reconstructed in this volume or only some? And this is more what is classically defined as a sort of dense connectome and sparse connectome. The other one is in terms of proofreading. And so even within a volume in which all neurons are reconstructed, some of them will be proofread to a higher level than others. Okay, and this might be by region or by part of a neuron. And so we always need to have that into account. And the other one is completeness of synaptic connectivity. So we know that the dendritic outputs, dendritic inputs to a neuron on these twigs are really fine neurites, and they're hard. Well, the autosegmentation algorithms don't segment these out right away. And so you need a lot of proofreading effort to actually connect these to the neurons. And so some of it will be missing. And so just very briefly, in terms of the main limitations or comparisons between a sparse and dense connectome. So a sparse would be something like the community that we have in FAFB, in CatMate. And so this is mostly manual or semi-manual reconstruction. You have to choose what neurons you're reconstructing, but then you can choose what level of completeness of these neurons you want. Okay, so you can complete them regionally. You can annotate synapses in just a particular area or not. So you have much more control. On the dense connectomes, these are the outputs of automatic reconstruction. You can have quality different between region depending on the quality of the image data in these very large volumes. So I know all neurons will be reconstructed to a certain extent, but you might not know without doing quite a bit of work what that baseline of completeness is. And then, of course, you can always, there will be some synaptic connectivity which can be validated. So you can get a sense of how it's representative of what it really is there. And just in addition to, so for example, in the workshop, you're going to be working with data from FAFB and CatMate and from the HemiBrain. And so you'll have numbers of synapses for the both. So they, depending on what is defined as a synapse, either by manual annotators in FAFB or by the algorithms that are used for synaptic segmentation, the numbers they can be compared, but they're not directly related. So 10 synapses in FAFB doesn't mean that we would expect 10 synapses from the HemiBrain data. Okay, and just to finish off, we have, just to mention that there are additional data sets already out there. So one is a female ventral nerve cord from Wei Li's group in Harvard. So there's also a tracing community that is tracing on there, and they will soon be providing an autosegmentation output and platform, FlyWire style, for people to trace. And so again, that will make it much quicker. And one data set that is not yet available, but will be by the end of this year, will be a male VNC. And this is part of our collaboration with Janelia, which we're already working on. And so at the end of this year, that should be. And so we'll then have a male and female VNCs for comparison. More in the future, we expect to have a male CNS, probably mid to late 2022, and the female CNS, maybe in 2024, that will be released then. Okay, and these will all follow the HemiBrain release sort of pipeline. In that sense, there will be probably a new print resource for people to get the data from there. And in terms of other advances that are worth mentioning, just to mention, Niels Eckstein from Jan Funke's group in Janelia has been working on predicting neurotransmitter from EM synapses. And so this has been done in FAFB. And so we have some, good impressions from what the method is doing. So that's really encouraging. And so there'll be a lot more work coming from this. Okay, and that is all from me. And I will pass it on to David.
Do we want to stop for any questions now for this section, or move on to David?
May I ask one question?
Yes.
I was wondering how easy or possible is the transfer of annotations or connectivity or shape? So for example, can you transfer information from the female VNC to the male VNC, or even, you know, from one species VNC to another insect, let's say? Is this possible? Or is there a way to have, you know, a fully annotated, or a well annotated or a well segmented data set and transferred some information?
So you can, I guess, you can transfer information in the sense of you can, I guess, plot neurons in different brains or in different template spaces. And so you could take a neuron that's been traced in the male VNC and transform it onto the female VNC template. And in that sense, you know, compare to whatever is there already. In terms of the other way that you can compare things is, for example, by using cell types. So if you annotate the data in one particular set of neurons with a neuron type, you could then compare it to neurons of that same type in another data set. Again, either bridging them onto that data set to see if there are any particular differences in morphology, and then looking at connectivity as well, if that's available.
Thank you.
I have a question. First of all, thank you so much for the presentation. That was really informative. I just have a technical question in terms of the completeness of a neuron that has been traced. In terms of when you work on, so I understand there are different level of completeness, but when you work on a neuron and trying to figure out the function of these neurons, how much percentage in your experience were you being satisfied with the, what I'm trying to say is, what did you know the connection is fully capture all the major connection of these neurons? How did you interpret it when you see other people reporting the trace of the neuron? What would be the critical thing when you determine whether that tracing is in a satisfied completeness range? Maybe the question is so vague, but I would just, whether you can talk a little bit more about in terms of how would you, when you work on the neuron and then tracing, what would you look for in terms of the completeness?
Mm-hmm. So I think there are a few different things. So one is in terms of knowing how complete a neuron is, for example, if you have several neurons in a particular cell type, that you look at how similar they are, or where there are, you know, big discrepancies. So that might indicate that neuron is quite incomplete, either morphology or connectivity. But that's so if you can have a set to compare. In terms of the work that they've done, even from our manual tracing initially, so we found that sort of sampling around 30% of, for example, if we go downstream, of neurons, if we sample 30% of connections, that would give us we would recover all the strong partners. Okay, so again, there will be a long tail of very weak ones, which will be, some of those will be random. So I guess we haven't really linked a particular number of synapses that we see in the EM to functional role. And so that we don't know. In terms of morphology, we do know that there can be cell types that can be quite similar to each other. And so the more complete you have them, the more it allows you to then connect those to either, light level tracings or GAL4 drivers or other driver lines.
Could I just chip in very quickly there? So I think that's a really important question for us as a field, actually, moving forward. What Vivian, was that right? Just us there. because we're going to see quite a lot of use of connectomics data over the next few years, and it's going to vary just how sort of complete the reconstructions were. One thing which obviously will be good is if you have the same neuron reconstructed in two different brains, you can just look whether the number of synapses, whether the amount of cable is similar. There is another thing which, if you go into and dig into some of our recent papers in March of last year, that the number of synapses sort of per micron of cable for a neuron when you've actually reconstructed it to completion seems to be actually a pretty, a rather stable number. Philip will remember that probably off the top of his head, but I think it's around 1 synapse per micron of cable. And so if you see reconstructions which have, let's say 5,000 microns of cable and 200 synapses, then that's about 25 times too low, based on that very rough handful, that very rough expectation. And so that might be something that you could just bear in mind when you look at data coming up in front of you. We should maybe move on to the next section, but those were all great questions right away, and we will collect more of them, and you're obviously free to use the work, the hands-on sessions to ask questions of that sort, not just coding stuff.
Okay, so that was a great introduction, Marta. This is not as much science, this is just about resources for integrating to make it easy to integrate this data. So Virtual Flybrain is a resource we've been working on for about the past 10 years that integrates data from a lot of different sources. This is just sort of showing the major sources of data integration that we've got. So we light level data from a lot of sources. We've got single neurons, including connectomics data from various different CAP-made databases, some of which we host ourselves, from FlyEM, Uprint at Janelia, so the HemiBrain that and FAFB that Marta's just been talking about. From the Flylight database, we've got GAL4 lines and splits. We've got lots of individual lab data sets that people have donated as well. And we're in the process of starting to link into the FlyCell Atlas, which does single cell RNA-seq. So actually, probably Nikos is going to be interested. The first one that we're integrating is the optic lobe, because that's really the most mature in the nervous system so far. Can
I actually, I just want to, this is a personal thing, but so, I think what David was just saying there, basically about linking, molecular data like single cell RNA-seq to connectomes, to me, that is the big applied problem in fly neuroscience over the next few years. And we'll try to, give you some pointers about computational tools that can help. But for our whole field, I think that's probably the biggest issue to making rapid progress.
Yeah. And I think in the central brain, it's especially hard, I guess, because you've got such small numbers of cells per organism. So the coverage you need to get, and then the understanding of the markers, it's much tougher than with the optic lobes. There's much larger numbers of cells, and this is Nikos could give you the details, and also quite a good set of splits that have covered them all. So from the bulk RNA-seq, from the splits, we've got quite good mappings to cell types. But yeah, it's going to be a lot slower with the rest of it. Maybe the peripheral nervous system, they're getting quite far with the ORNs now, for example. Okay, so we do two types of image integration. One is we do two types of data of integration. One is image-based, where we have data from many different sources and cross-registration to bring everything onto a standard brain volume, and that links it to a standard parcellation scheme. So Greg's actually been involved in a lot of this work from the start in this field to create all the bridging registrations, for example, and work on bridging. So that allows us to integrate data both visually, so you can inspect it, but also so it's amenable to common algorithms to analyze it. And the other type of integration we do is semantic integration. That semantic integration works around this, the Drosophila anatomy ontology. Claire is on the call here, is the main curator on this now. So this is a pretty comprehensive now, I mean, as far as current state of knowledge of the field, of curated queryable classification of Drosophila neurons and brain regions of types of neuroanatomy. It gives us a standard nomenclature, standard symbols, synonyms from lots of different papers in the literature and references. And this is extensively linked to 3D images and connectomics data. So in terms of the terms that we have, we've got over 8,500 neuron types now, actually, so 3,700 of those are provisional ones that have come from the HemiBrain project, but almost 5,000 of those are curated from the literature. We've got over 1,300 nervous system regions, so that includes things like tracts as well as synaptic neuropil. That should say 16,000, not 6,000, sorry. Anyway, we've got a large number of queryable statements which kind of make this kind of into a tractable database that you can ask questions of. And I'll give you some examples of that later. We use this Drosophila anatomy ontology to annotate large numbers of images. We've got, typo again, it's 117,000 images, not 177. That includes 67,000 single neuron images, covering 5,000 different types, and nearly 25,000 of those with associated connectomics data, and that's climbing all the time with the pipelines that we've built to various other databases. We've got very large numbers of expression patterns, including 4,500 images of splits covering 600 combinations now. So that includes all the Janelia splits, but also things coming from other sources as well that people have donated to us. And we've got extensive curation of where drivers are expressed. So for example, for the splits, for 890 split combinations, so for more than we have images for, we have over 2,000 statements about expression that links them to 464 neuron types. So I think it's been able to jump back and forth between the splits and the neuron types is really useful to the field right now. And we've also got links into ways to predict which neurons are in which splits as well. So the general nomenclature problem, different resources use different names for neuron types and for the sort of general classes that you might use for neurons as well. So how do you take information from the literature, like an assertion about where a split line is expressed, and how do you take all of the different information in different databases about images and connectomics, etc., and integrate that into something you can cross query. So that's really what what we're trying to solve with all this curation and with the web app that we produce. So here is a little screen cap of the Virtual FlyBrain web app. You can here we've got 2D and 3D images of some neurons and split expression patterns, and some details of a particular neuron and its classification here. And we can run various types of queries and searches. So I can search with any name in the literature to try and find a neuron of interest. And we pull in names that are used by databases as well. So with a reasonably sophisticated autocomplete search, it should be reasonably straightforward to find the terms that you're interested in. And then once you find them, there's additional information and links to the literature, images, etc. You can browse around as well. So you could start from that specific term you found and maybe look for a more general term that you might want to use to query across different data sets. So maybe you wanted to look for all auditory system neurons, for example, in a particular part of the brain. And you can drill down as well. So you could take a term and you could drill down to more specific terms here. So here's a DA1 neuron, and here's a couple of subclasses of that. And we have more sophisticated types of queries. So this is a query for, jumps around a lot, for dopaminergic neurons with presynaptic terminals in the adult mushroom body and postsynaptic terminals in the superior intermediate process cerebrum. So this, we can now do these kinds of things with with across data coming from the connectomics. But I'm not really going to go into much of the. Oh, and here's an example of a result here. I'm not really going to go much into the types of queries that you can do on VFB because just finally, we also have integrated queries by morphology using NBLAST algorithm, which was actually developed by Marta Costa and Greg Jeffries here. So I could take a single neuron and I can get a query for neurons with similar shape and get a similarity score back a little bit like BLAST. But what we're really interested for this workshop is facilitating programmatic usage. So we have an API. In fact, we have a couple of APIs. We have a REST API but we're going to concentrate here on a Python API that we've developed, which is more fully functional right now. And that allows us to take, for example, the identifiers that are used in different databases. So you could query on VFB, pull back a list of neurons, and then convert that into identifiers that you could use to search Newprint, or convert that result into identifiers you could use to search the CapMate API. So FAFB, for example, or you can go in the opposite direction. If you're already in one of these APIs, you can get a set of IDs and use that to pull information from Virtual Flybrain, or to pull information from Virtual Flybrain and then bridge across to a resource like Newprint. So that's really just all I wanted to cover. It was just a really sort of general overview of what the project is and why it's kind of potentially useful to the effort of trying to query across all these growing number of databases that we have. I mean, I think actually for the adult brain, things are a little more unified. You can get quite a bit further without an integrator, but that's not true of the whole nervous system. So certainly not right now. And it definitely gives you a lot more power to be able to do the kind of abstracted queries. So if I just wanted to say, give me all the auditory system neurons, for example, that's not something you could do in any of the individual resources. And okay, so this is just the team. So on this we've got Robert, Marta, Claire, and Alex are all on the workshop with me. So if there are no questions, we could move on to the sort of to Philip's presentation of the API landscape.
Okay, so this should be really short and sweet, and it's meant as a segue into the hands-on, which I'm sure you're anxious to start with. But we thought it'd be good to, basically briefly introduce all the tools that you will cover and learn or explore in the hands-on bit of the workshop. And since this is split into an R and Python part, so is this this document. By the way, this is just a markdown in a GitHub repository, which I think, David, correct me if I'm wrong, this is going to be shared with everyone, right? The GitHub repository.
We could already put a link on
it's public.
Yeah.
Okay.
We could put a link on the agenda already.
We should put a link on the agenda. Yeah, it's they're sort of indirectly connected into it because all of the projects have it. But yeah, let's do that.
Yeah, I think all the workshop notebooks will eventually end up in here if they're not already in it. Anyway, so this markdown should be sort of a nice hub for you if you want to later on go back in and whatever look for something that you can't quite remember the name of. It has all the links, so what I'm telling you right now is already spelled out somewhere in the text. Anyway, so again, our part, Python part, David already alluded to that on the querying the VFB API and databases. We've got on the Python part a package called VFB Connect, which is, I think, one of the first things we'll get hands-on in the workshop. And then for R, Greg principally, I think, wrote a little wrapper, which is really just sort of, if you know Reticulate, it's just using exactly that. So there's, I think, a one-to-one correlated functionality. Then when it comes to working with neuron data and querying the various data sets, for example, FAFB via CapMate or Newprint containing the Genea Hemibrain, there's a range of different packages, but they're all interconnected. So they all sort of work nicely together. On the R side, we've got the so-called netverse that's sort of analogous to the tidyverse. So if you're familiar with art and you know what I'm talking about. So this is a collection of packages that are sort of your one-stop shop for all things Neuron. It's been principally developed by Greg, and at least the base package net has been around for how long now, Greg? 8 years, something like that.
Yeah, I mean, with a bunch of collaborators from the lab over the years.
For example,
including Mata and Shri, who are both on the call.
So it's been principally developed by Greg, but as he just pointed out, Marta, Costa, and Sri, who are both on the call and will be some of the supervisors for the hands-on workshop, are also here. Right, so we've got this netverse, which contains a whole collection of packages. At the core of it is net, which is sort of focused on neuroanatomy, but you know, sort of has expanded out of this sort of initial, I think, purpose. So this is basically whenever you want to work with neuroanatomy, that can be analysis, manipulation, NBLAST, transforms, you name it. Then to query the NuPrint database, there's 2 packages actually. One is called NuPrinter. That is to make queries against the NuPrint database. And then there's HemiBrainer, which is has been principally put together by Alex Bates, a former PhD student in the lab who's since moved on, actually fairly recently. That contains a lot of metadata on neurons in NuPrint. And then to query CatMate, use RCatMate. Then on the Python side, there is sort of the analogous package analogous to Navis, which has been principally developed by me. For querying CatMate, you have PyMate, which is built on top of Navis. So basically, if you pull stuff via PyMate, you can plug it directly into Navis. And I should probably mention that's also true for RCatMate and NuPrinter. So basically, data you receive via those packages works with the base package, whether it be Net or Navis respectively. And then to query NuPrint, we will use a package called NuPrint Python, which is the Python library that has been is sort of provided by Janelia to query the NuPrint database. Just sort of a quick mention, I don't think we'll cover these in the first day of the workshop, but since the second day is a bit freeform, as Greg mentioned, we might, you know, who knows, we might end up getting to those. I
intend to.
NBLAST, which lets you compare neuromorphologies, right? So this is extremely useful if, well, A, you want to find the same neuron across data sets, or if you want to cluster neurons to, say, produce cell types. You can do this with net.nblast on the R side and with Navis on the Python side. And then the second bit that is sort of bread and butter for us, really, is transforms, which means, and we've had a question for this earlier, basically lets us transform spatial data between template brains, right? For example, between whatever, the FAFB data set to the HemiBrain. And then I guess in combination, those two are really becoming powerful. For the transforms, you would use net.template brains, net.fly brains, and net.jrc brains, which contain different kinds of data and different transforms. And on the Python side, there's fly brains. Unless there's any question at this point, I would just wrap it and say we move on and get our hands and feet wet. Hands dirty, feet wet.
Okay. Any questions for Philip, or indeed any final questions across those three?
I have a question. So is there any advantages of one of the platforms on the other one?
Maybe I'll take that.
Okay.
Okay. So I think the biggest difference right now is basically the language Python versus R. I started developing these tools quite some time ago. There is still even a little bit of code from my PhD when I was with Li-Chun Lo at Stanford, buried deep inside the NAP package. I think it's a very mature set of tools now. Sometimes it can be hard to find what you're looking for, and that's one of the things that I hope to orient people to during this, during this section. I guess I think honestly that's the biggest difference right now. is just the choice of language. There are certain pieces of functionality that might be a bit more mature in one or the other of these tool suites, depending on when Philip or I have got round to implementing new stuff, or occasionally other people. But they're actually, even like the sort of the overall design is quite comparable. So most of what Philip has written was written sort of, after looking at stuff that I'd done, and he sort of took, the best bits of that and then sort of tidied them up. So there's some sense in which, Philip's, I quite like the organization that Philip has at this point, because he's been able to look at some of the, not too bad, but the occasional mistakes that that I made. So if you had to choose between one or the other, I think you should mostly just choose on what you're comfortable in terms of language, and there will be very little functionality that's missing. The main thing I would say right now is that I think there are some basic operations that are a little bit simpler to write in R, because we've kind of made we've made sort of little shortcuts to make certain kinds of things easier. But if you don't know either language, I think it's fair to say that more people are using Python than R overall in scientific computing, although in genomics, I guess Python is still, R is still, very popular and perhaps more represented. I don't know, Philip, I think given that spiel, if you have any extra.
I think that's a really good wrap up. Maybe just to add that if you know that you want to use some third-party library, and it's only available in R and Python, that might sort of help you make the decision. That being said, there's wrappers both ways, right? So I just mentioned the reticular package, so you can, it's fairly straightforward to run Python libraries or use Python libraries from R and vice versa. Probably actually at this point easier to run Python libraries from R than the other way around. But if you have anything where this is going to be sort of a rate limiting step, and that might just sort of tip it in the favor of one or the other language.
Yeah, I mean, we're definitely using both now, pretty regularly. So most of the people in our group will use both. People typically will gravitate more to one, or to use more one or the other. But certainly, there's plenty of functionality now in R where we've wrapped an external Python library. So it's quite efficient, and you'll actually use that in the workshop. So it's certainly quite efficient to do that. As Philip says, it's slightly clunkier in the other direction, although that it can work too.
So just to add on a point there, from what I found from using both R and Python, is almost all of the standard analysis like that you would see in any paper for connectomics that is already available in the R package. And I switched to Python only if I wanted to do something new. And I saw that there are libraries available that are easier in Python, then I would simply switch to Python to do that part of the implementation alone.
Any other questions before we start to break out?
Hey, Philip, do you have plans to integrate some of the Navis or PyMade packages with some of like the mesh analysis tools that like Casey and them at the Allen Institute have written? I mean, maybe you have other solutions for working with meshes, but given the new segmentation data, I'm just curious what your thoughts are there.
We actually had a chat about this a couple of weeks back, and so the issue is that they've sort of in parallel come up with their own ecosystem, and they've approached it from a different angle, in the sense that we've historically been working with skeletons because we've been doing this manual reconstruction, and they've done automatic segmentation, so they've come at it from a sort of mesh perspective. And I guess at this point we're starting to meet in the middle. The problems that already both us and them are sort of already invested in our respective ecosystems. I guess my plan is to probably do the same thing that I did with Greg's R libraries, which is plug the things I like and re-implement them. That might ultimately be easier than trying to merge these by now fairly big projects. Cool. But yeah, if basically I think I speak for both me and the others who are actively developing those tools, we're generally very open to feature requests. And if you think something's missing, or if you find a bug, all of these projects are open source on GitHub, and we're generally really happy if people use it and have interest in it.
So yeah, I mean, just a couple points to add there. So Brandon is in John Taltill's lab, and I guess the people he was referring to are in the Allen Institute, working with mouse data. Obviously, the data types that we're working with are all like that. There's not that many different data types to work with. But there are quite a few connectomics toolkits available, or neuroanatomy / connectomics toolkits available right now. That said, I would say that probably the ones that have been written in our group are probably the most extensive. And that's partly because there's such a range of different stuff you can do with Drosophila neuroanatomy data at the moment, especially in linking, for example, light level and light level and EM connectomics data. So to my perspective, certainly if you're going to work with flies, I think starting with what we're going to teach you this afternoon, this morning, depending where you are, is really the place to start. And one other very quick comment there, in terms of, Marta mentioned in her talk this difference between mesh and skeleton data. So hopefully you'll get more of an appreciation for that during the hands-on, but it's actually a really significant difference. So, mesh data, you can get nice, really nice morphologies. You can see the presynaptic boutons on neurons, and that's great. But there are lots of times for analysis where you really want to know where on the, branching tree of a neuronal arbor is a synapse. For example, if you might want to split it into an axonal or a dendritic arbor, something that we hopefully will get onto tomorrow. And that will normally require a skeleton representation. So the challenge right now is to merge those two kinds of data into, so that you can fluidly work with either, depending on what you need to do. And actually, Casey Schneider-Moselle, who used to be in Albert Cardona's lab, has a very good approach to that, which I'm sure Philip will be borrowing soon.